Hi guys, Anna is here, and in this video we're going to be working through the protein structures that you need to know for A-Level Biology. So please grab a piece of paper, and without further ado, let's get into this topic. So in my previous video we discussed that the proteins are the polymers of amino acids, and in this video we're going to learn how that polymer is shaped into a 3D shape that can be used for different functions. So this topic can be subdivided into 4 subtopics of primary structure, secondary structure, tertiary structure, and quaternary structure. And this will be basically the plan of the video. And as long as you can differentiate between those structures, then you can understand the overall topic. So let's start with the primary structure. So the primary structure is formed by many amino acids bonding together via the peptide bonds. If you don't know how this happens, please watch my previous video, which I'm going to link in the link above. So let's go and draw this. So I want you to draw a couple of circles like this, which each circle will be representing amino acid joined by peptide bonds. Okay, here we go. So what you guys need to remember is that the primary structure is polypeptide, it's what it's called, and by definition, polypeptide is the chain of amino acids, the straight chain we've just drawn above. Okay, so the polypeptide is going to have a carboxyl end at the end, or sometimes it can be referred as carboxyl terminus, and on the other end there will be an amino end, or otherwise referred to as amino terminus. Polypeptides are synthesized at the ribosomes, where translation happens, which are usually attached to the rough endoplasmic reticulum. And what you might have not known is that when it's a straight polypeptide, it's actually taken inside into the ER, and that's where it becomes folded protein. And for the rest of this video, we're going to now discuss of how this protein becomes folded, because the folding will give it 3D shape, which is essential for its function. Okay, let's jump onto the secondary structure of the protein. So the secondary structure will provide an extra complexity to a protein. So the initial folding, there are 2 separate structures you need to know of, which is alpha helix and beta pleated sheet. What I would recommend is just by checking the images online as well, because I can't draw them very easily using the pen. But basically the alpha helix is a folded form of a secondary structure, and beta pleated sheet is the things running kind of as a squiggle and in parallel. So it gives this extra complexity to a protein. So this is due to the presence of the hydrogen bonds between different amino acids. Hydrogen bonds are intermolecular forces which give extra structure to biological molecules, but in this case they will be occurring between different amino acids of the amino end and the carboxyl end. The placement of hydrogen bonds depends on the order of the different amino acids and where they are within a protein. After creation of the secondary structures, the protein will be further folded to form tertiary structures, which we'll discuss in a moment. But the tertiary structure is essentially a 3D shape of a protein. You need to guys understand that the shape of the protein is very important because the proteins have so many different functions. So let's say we have this protein now that has been slightly folded, and I usually draw a 3D shape of a protein as this squiggly line demonstrating the complexity of a 3D shape. And the functions include enzymes, receptors, hormones, and so many more things. So for any specific functions, the proteins must have a specific shape. There are extra 3 different types of bonds that you need to know to understand how this 3D shape is created. So let's start with ionic bonds. Ionic bonds are created between the amino and carboxyl groups that are not involved in the formation of a peptide bond. This means that these ends, the amino and carboxyl groups, they can come from maybe the end of the polypeptides, or they can come from the variable groups called R. The second type of the bond that can be involved in the forming of the tertiary structure is disulfide bridges. This can only happen between 2 cysteine amino acids, because cysteine amino acid has sulfur as an element in its R group. So if we just draw it out, it's going to be CH2S and then another S, CH2, and between those sulfurs, the disulfide bridge can be formed. So it's unique to those 16 amino acids. And the final bond that results in the formation of the tertiary structure is extra hydrogen bonds that will be present in the structure. So in summary, the presence of these bonds in a different order depending on the order of the amino acids results in the unique shape of a protein, which is essential for its function. Okay guys, this is the end of the video. In my next video, I'm going to be discussing the quaternary structure using hemoglobin as an example. Please press the thumbs up for this video. Thank you so much for your support already. The more subscribers I get, the more videos I can make. And I'll see you in my next video. Goodbye.